בסדר? היי לכולם, אני רבית, ההרצאה היום תהיה באנגלית, בגלל שזה מועלה אחר כך ללינקדאין, פשוט חשוב לי. So my name is Ravit, and I have a PhD from the Faculty of Medicine in the Technion. I graduated from Primrose last year, and ever since then I've been doing projects both in academy and industry. And that to say, I'm open to work. My talk today will be about AI and generative adversarial networks in drug design, and I hope you will enjoy it. So let's begin. Today's discussion will include AI in healthcare, a little bit of background, advantages, risks, and challenges, and also give a successful example of implementing AI in clinical care. I will talk a little bit of GAN principles, mathematics, a little bit loss functions, training process and challenges. And also I will talk about a publication that was published in Nature, where the authors implemented GAN in generating new molecules. So why use AI in drug design? First of all, it's a new approach for discovering new drugs. And as some of you or all of you may know that clinical trials and preclinical trials take a long time and can be very expensive and with very low success rates. AI in drug design is to save time and costs and to increase the success rate of preclinical and clinical trials. There are challenges in implementing AI in drug design. First of all, you need to obtain an appropriate data set. You have to have a large scale size of data set, correctly annotated data. Also, you have to understand the context between the data and its outcome. If I'd like to give you an example. For instance, in preclinical trials, the outcome may not be the same as the outcome in clinical trials in animals. The preclinical trials in animals may not be the same as the outcome in clinical trials in humans. And also, biology field is rapidly evolving. Ontology is constantly changing. This may affect the correctness of annotated data. And mistakes in data reports may skew predictive models. Another issue is imbalanced data. For instance, in literature, you would normally find only positive, compounds with positive activity rather than compounds with negative activity. And this makes it difficult to obtain balanced data from datasets. And this is an example for early detection of breast cancer with AI. During the pandemic, in a general hospital in Massachusetts, experts noticed that patients tend to either delay or skip routine checkups for breast cancer. And this compromised treatment and proper diagnosis. And they decided to implement a generative adversarial network-based algorithm developed in the lab of Regina Barzilai in MIT. And they compared to an existing statistics model, and they saw that 42% of people who went on to develop cancer in 5 years were flagged as high risk by the algorithm, compared with 23% for the best existing model, which is, I think, quite impressive. In 2014, Ian Goodfellow published his article, Generative Adversarial Nets, or in short, GANs, which instigated the use of GANs in deep learning. I will discuss GANs shortly right now. GANs are comprised of two networks, generator and discriminator, and both compete in a zero-sum game. The generator receives random input with noise and generates fake samples. And then the fake samples are fed into the discriminator with real samples. The discriminator's job is to discriminate whether the samples given to him are either real or fake. And this is a zero-sum game. If the discriminator fails to discriminate real and fake data, then in the next round, the discriminator is penalized and the generator is rewarded. If the discriminator succeeds in discriminating correctly, then in the next round, the generator is penalized and the discriminator is rewarded. And eventually, the discriminator and the generator reach equilibrium in which the discriminator discriminates correctly around 50% of the time. This is in ideal cases, as I said. In the next slides, I will say why it's ideal and why in reality it usually doesn't happen. This is GAN loss function minimax. Basically, the discriminator seeks to maximize the log probability of discriminating real data and the log probability of discriminating the inverse of generated data, both sides of the equation, and the generator seeks to minimize the log probability of the inverse of generated data. This is the training process. This graph is taken from the article of Goodfellow. The blue dashed line is the discriminator distribution. The black dots are real data distribution, and generator, the green continuous line is the generator's distribution. As you see, as we move along the process, and arrows below are the domain from which the Z vector is sampled from. As we move along with the process, eventually the generator distribution is very, very close to the real data distribution, and discriminators, as I said, discriminator discriminates correctly around 50% of the time, only in ideal cases though, because GANs are really hard to train. Why are they so hard to train? There are three scenarios in which GANs can fail. One of them is mode collapse, in which the generator generates a small subset of samples. If we talk about images, then the generator will generate a repeating subset of a small subset of identical images. And also vanishing gradients. If the discriminator is too good, then the generator fails to generate new data and the weights don't get outdated. They become very, very small. And you'll also have to remember that we are training two networks all at the same time, and this is a really hard thing to do. And in many, many cases, the GAN models do not converge. I talked about the minimax loss function. There are other loss functions, but I will talk about two because they use it in the publication that I will show you next. One is a Wasserstein GAN, in which the discriminator is replaced by a critic, which job is to score the realness of the data. And the conditional GAN is a GAN in which the generator generates data with a condition to generate a certain type of data. In this publication in Nature, they implemented GANs for generating heat-like molecules from gene expression signatures. Before I go deep into this article, some of you may not be familiar with the biochemical terms, so I will talk about them a little bit. One is the simplified molecular input line entry system, or in short, SMILES, which is an ASCII string representation of a molecule which describes the structure of a chemical species. You can see in the picture here is an example of a SMILE of ibuprofen. Other terms are heat molecules. which are compounds with desired type of activity, and gene knockouts, an alteration in DNA sequence which is translated into non-functional protein. And measurement methods they used for chemical similarity, they use three methods: MAX, molecular exosystems, similar chemical groups; fragment similarity, similar molecular fragments; Morgan fingerprints, a vector representation of the molecules. Just three types of measurements. This is the model itself. They used two stacked GANs of the same time. They used conditional Wasserstein GAN with penalty. The first GAN received input with random Gaussian noise, and the output of the first GAN was fed into the second GAN. The second GAN was actually a refinement of the first GAN. This was to achieve a more accurate results. Here in the loss function of the generator, the F function job is to calculate the probability of the compound to be from a certain type, here as well. And alpha and gamma are regularization parameters in this model. This is the similarity evaluation between generated and real compounds. They calculated the similarity using mean and covariance of regular distribution and mean of covariance of generated compounds. This is the model pipeline. They took smiles and they fed it into an encoder to achieve a latent representation of this compound. And as you can see here, as I said before, the model is comprised of two GANs, two stacked GANs. The first one receives gene expression with Gaussian noise, and its output is fed into the second GAN, which is a refinement of the first GAN. This is the general pipeline. They took 20,000 compounds with gene expression signatures, and they fed it into, they took compounds from the L1000 dataset, and then they generated with the model new molecules, of which 10% were viable and synthesizable for synthesis in lab. In this graph, I would like to show you the fact that the addition of the second GAN elevated the accuracy of this model, increases the accuracy of this model. Here you can see that in the upper graph, the blue line represents molecules generated in the first GAN here. And the green line represents the molecules generated in the second GAN. As you can see here, the second GAN had higher density of unique and valid molecules, which means that adding a second GAN to this model increases the accuracy. And here are examples of generated molecules and the reference molecules, compounds, and the closest reference compounds based on the similarity. This model was also shown to be effective for optimizing compounds. Here in this graph, they took a benzene compound and they fed it into the encoder, and they got encoded the scaffold. And then they fed it as input to the second GAN, and then they got optimized molecules based on the benzene structure. As you can see here below, inside the circle, these are molecules generated after optimizing the benzene structure. And outside of the molecules are reference molecules with known activity that are the closest to, in similarity to the ones inside the circle. So they compared this method to other methods. For one thing, they generated molecules with this model and compared it to other generated molecules. And they saw that the similarity and the density of similarity in all three measurements is much higher in compounds generated by this model compared to other models. And also they compared this model to non-conditioned GAN and non-conditioned LSTM, and then they showed that classification score for the conditioned GAN is much, much higher than the other two methods explored. They concluded that the model has advantages. First of all, conditional GANs are an innovative and improved approach for generating new molecules from a particular gene expression signature. They are also able to extract all information contained in compounding gene expression data, in particular the one in L1000 database. Also, no previous measurements of biological activity or target annotations are required, which is great because this means that the model is applicable to many targets, not just the ones they tested in this article. And also, as we shown on the benzene ring, the model can be used for compound optimization. However, there are challenges because there is variability of drug responses in different cell lines. And also, further research is required for generating molecules with non-structural features, with activity on specific drug targets. So this is it. I hope you enjoyed my talk today. And if you have any questions, you're more than welcome to ask me. Thank you.
Yes, if I may, I have a question. First of all, thank you for the very interesting lecture. I would like to know, I understood how the GAN works in terms of finding viable molecules, but how does the GAN generate, how do you label which compounds act on which diseases or which medical conditions? I didn't get that.
They didn't go as far as that. They generated molecules and then they compared the molecules to reference molecules with active to compounds with known activity. And the main goal was to my understanding, was to generate new molecules that are synthesizable in the lab for further studies.
Okay, so you find something that's synthesizable, and then you see if it does something.
Normally, that's how it works most of the time, because first you have to generate new molecules, new viable molecules, and then after that you can continue to further studies in different aspects.
Good, thank you.
You're welcome.
You mentioned this was published recently. Can you elaborate where it was published?
In Nature. I don't remember the year, but here. Where is it? Here it is. Oh, sorry. There you go. Nature Communications.
Thank you.
Any more questions?
Not maybe some comments from a more like a practical point of view. We know that there are some companies that are doing this kind of work, like Benevolent AI. If you heard of them,
No, but I will write them down right now.
They're doing really beautiful work in drug discovery, drug design.
Here in Israel or
No, in the UK.
in the UK?
So what I think the challenge here, because this kind of loss function is something that everyone now knows how to do it, right? So what is the competitive edge that a company can have? So the competitive edge is, at least that's what they do in benevolent AI, is how to extract and parameterize all the knowledge and the information that exists in the literature. And this they do by very, very powerful NLP tools. And this is because if you know how to extract everything from the literature, then you have tons and tons of information, and you can build a huge learning set from the literature.
You're
so
talking
this
about text
I
from
think
articles?
yes, what they do is they just look for similar compounds in articles automatically, of course, and try to see what they do. If they do similar things, maybe you can exclude them. Maybe they have all kinds of bioavailability problems and that kind of stuff. And one more important point here that I think is important when you try to use AI in pharmaceutical industry is the problem, the explainability problem of neural networks. Because of the abstraction of neural networks, it's very hard, because regulation today requires that you explain why you did this or that. So when you use like abstract tools like neural networks, it becomes a challenge. And this is why many works in this field using AI in pharmaceutical development use decision trees, which is like a kind of a primitive way to do it, but they are explainable. This is I think an important point, is how, but we can see if you look at the benevolent AI website, they do have some compounds that are advancing in like in advanced stages of development, and they apparently they know how to explain that to the regulation. That's it.
Interesting.
Thank you. Very interesting work. Yeah.
Thank you. Any more questions?
אוקיי, נעצור את זה.